Well, hello everyone. Welcome to an overview of micro and bacteria. I hope you have your coffee, whatever you have. I hope you're enjoying it. So today what we're going to look at is bacteria in general, and it's important to understand the different kinds of bacteria because depending on the bacteria that someone may have become infected with, we have to provide them a very specific antibiotic to fight it. And normally we can't do that unless we take their blood and run a blood culture, right? So types of gram-positive are Staphylococcus, Streptococcus, Enterococcus. Gram-negative cocci, Neisseria and Moraxella. They're kind of pretty, aren't they? Different colors. So gram-positive rods, we have Listeria, Actinomyces, Corneum bacterium, Nocardia, Mycobacterium, Bacillus, and Clostridium. So the site is where uh, we have common organisms that may or may not be commensal. The blood is obviously sterile. The cutaneous surface or our skin, we have staph epidermidis. Less common is Staph aureus, Corneum bacteriae, Streptococci, and yeast. So keep in mind, we can't live without bacteria, okay? We need bacteria. As many times as you wash your hands, the best that we can do is medical asepsis. We're never going to get rid of everything, okay? There's always going to be organisms on us. After all, we are just one giant organism. There was a study, I can't recall how long ago it was, but they said that the amount of bacteria in our body exceeds that of cells, if you can believe that. Do some research. In the nose, we have colonization with Staph aureus, Staph epidermidis, Corneum bacteriae, and Streptococci. In our mouth, or the oropharynx, we have Streptococcus and Strep viridans. The non-pathogenic Neisseria, Haemophilus influenza, and Candida. Gingival cerevisiae, we have anaerobes such as Bacteroides, Prevotella, Fusium bacterium, Streptococcus, and Actinomyces. Stomach, none, because our stomach has a very high pH, unless, of course, you get Heliobacter pylori. And Heliobacter pylori actually makes its own protective zone by secreting urease. It's like a capsule, and urease is very alkaline, as opposed to the stomach being very acidic. So Heliobacter or H. pylori is very hard to eradicate. You usually have to do a triple therapy with that: clarithromycin, omeprazole, and amoxicillin. The colon has microaerophilic and anaerobic bacteria. Babies breastfed only, bifidobacterium. Adults, Bacteroides and Prevotella. Babies can have lactobacillus and streptococci. Adults can have Eubacterium, Fusium bacterium, lactobacillus, and gram-negative rods, whereas the vagina is colonized by lactobacillus and assorted strep, gram-negative rods, diphtherioids, and yeast. And that sounds pretty scary, but the vaginal vault actually has to have uh, a particular pH where these can thrive, and they actually can help us fight off other bacteria. So organisms with capsules. You can memorize the mnemonic. Some killers have pretty nice capsules. I've seen it where some killers have pretty nice cars. Uh, these are the encapsulated organisms: Strep pneumoniae, Klebsiella pneumonia, Haemophilus influenza, Pseudomonas aeruginosa, Neisseria meningitis, and Cryptococcus neoformans. Let's take a look at this drawing of a bacterium. We have a capsule, we have a cell wall, we have a plasma membrane, we have a cytoplasm, we have bacteria flagellum, We have nucleoid circular DNA. We have ribosomes and plasmids and pili. Now, when we compare and contrast gram-negative and gram-positive, we'll see that gram-negative and gram-positive share some of the same anatomy, but their outer capsule's a little bit different. Gram-positive bacteria have a peptidoglycan layer. It's very thick, which is why it stains violet, because the thick layer holds the stain. Whereas gram-negative doesn't have a very thick layer, so it doesn't hold the stain, so it turns kind of a red. My niece, your legion, lists Francis, Bruce, and Salmon. These are facultative intracellular, which means that they live within the cell. Mycobacterium TB, Neisseria, Yersinia, Legionella, Listeria, Francisella, Brucella, and Salmonella. Some of the toxins. The endotoxin is equivalent to the lipopolysaccharide capsule. So when you think endotoxin, think N for gram-negative, because it's the lipopolysaccharide capsule that makes gram-negative bacteria kind of nasty. Also, the lipid A portion is the toxic part, and it has a tendency to overactivate the immune system, which can cause septic shock. In gram-positive bacteria, but you can have exotoxins in gram-negative or gram-positive with AB toxins. B binds to a receptor, and A is active, and cytolysins lyse the cell. So this is kind of interesting. This gives you the toxin mechanism, tells you the organism that does it, tells you the toxin, the mechanism of action, and the role in disease. Now this is important because this is how we determine what antibiotics are going to be used for a particular infection. So protein synthesis inhibitors would be organisms like Clostridium diphtheriae, Pseudomonas aeruginosa, Shigella dysentery, enterohemorrhagic E. coli. C. diphtheria has the diphtheria toxin, and you know that children have to be immunized against this. It's very deadly if a child gets diphtheria. The diphtheria toxin has its mechanism of action is the elongation factor 2, and it works on the heart, the nerves, and the epithelium. It also inhibits the eukaryotic protein synthesis. We're eukaryotes. Bacteria are prokaryotes. They don't have a nucleus. P. aeruginosa has an exotoxin A. So aeruginosa, exotoxin A. It also has a mechanism of action, which is the elongation factor, particularly dominant in the liver, and it also inhibits eukaryotic protein synthesis. Shigella and E. coli, these guys kind of lie on the same spectrum in the sense that they cause diarrhea and vomiting. If you ever had an E. coli, you had raw hamburger, raw meat, that's actually E. coli 057H7. But in any case, the Shiga toxin and the varotoxin associated with E. coli, which is also a Shiga-like toxin, interferes with the 60S ribosomal subunit. Now, most of the antibiotics we have that interfere with ribosomal subunits are usually the 30S and the 50S. Some of these bacterial diarrheal diseases we don't even treat with antibiotics because it can actually make it worse. Neurotoxins, tetany, everyone's had their tetanus shot hopefully, is a tetanus toxin. It causes spasticity and it causes the jaw to lock and muscles to tighten. They get that sardonic grin. It blocks the inhibitory transmitters of GABA. It's actually a reverse, travels in reverse. Um, and the amino acid that it uses is glycine. It inhibits neurotransmission in inhibitory synapses. So it basically disinhibits the inhibitor. Or, yeah. C. botulinum, very dangerous, causes flaccid paralysis. Maybe you've heard of floppy baby syndrome. That's why we don't give infants honey until their enteric system has developed enough immunity and bacteria in itself to be able to fight against the bacteria in raw honey. Especially honey that is not pasteurized and it's raw, you can't really give it to babies because they could be very susceptible to the botulinum in it. It blocks the release of acetylcholine at the receptor level, and we need acetylcholine to be able to propagate the neurons for movement, etc. And it inhibits the cholinergic synapses. Cytolysins like Clostridium preferengens and Staph aureus use the alpha toxin. You notice both do. The C. preferengens uses lecithinase to lyse these cells, and Staph aureus intercalates, forming pores. So with Clostridium preferengens, it damages cell membranes and causes myconecrosis. You can get what we call gas gangrene with Clostridium preferengens if it's under the skin. It almost sounds like crepitus. It's like crackle and pop right under the skin because of the lecithinase. And with Staph aureus, the cytomembrane becomes leaky. So here, looking at the toxin mechanism of superantigens, those bacteria that possess superantigens are Staph aureus and Strep pyogenes. Um, a toxic shock syndrome 1 toxin for Staph aureus, and exotoxin A for pyogenes. It is a superantigen. Strep pyogenes is similar to the toxic shock syndrome 1. It can cause fever, shock, capillary leakage, as can strep pyogenes cause shock, capillary leakage, as well as cardiotoxicity. E. coli has a heat labile toxin. It uses adenylate cyclase by adenosine diphosphate ribosylation of guanosine triphosphate binding protein, causes secretion of fluids and electrolytes, causing profuse and watery diarrhea. Vibrio cholera kind of does the same thing. So I'm glad they kind of lumped these two together because trying to memorize the mechanism of action can be a little bit challenging. So enterotoxigenic E. coli uh, is adenylate cyclase ADP ribosylation of GTP. Everything here that you see from E. coli down to pertussis uses cyclic AMP. Bacillus anthracis, or the anthrax toxin, it's a capsule made of a polypeptide. Um, elongation factor or edema factor, lethal factor and protective antigen is what is the mechanism of action that keeps anthracis in the body and very hard to kill. So the edema factor, it decreases phagocytosis and causes edema and kills the cells. Pertussis is also, if you recall, is a vaccination that children get. The pertussis toxin also ADP ribosylates protein G1, and it releases a histamine sensitizing lymphocytosis islet cell activation. So lymphocytosis, lots of lymphocytes. So here's the difference between gram-negative and gram-positive. So because of the big peptidoglycan layer, it stains purple because it doesn't wash off. Once we wash the slides, With gram-negative, it has a thin peptidoglycan layer and it stains pink because the um, the pink stays because the violet got washed off. So when we look at endospores, such as Bacillus and Clostridium, um, their function, as with all bacteria and spores, is to survive. So the vegetative cell has reduced nutritional conditions. They produce sporulation with loss of vegetative cell. So you have a spore and it can survive for years. If I recall, there was some research done. They had found a woolly mammoth in the ice, and the spores and the bacteria, well, mostly the spores, can lay dormant for a very, very long time, more than years. The core is formed by DNA, ribosomes, and glycolytic enzymes. We have a cytoplasmic membrane. We have a spore wall, which is peptidoglycan. We have a cortex, which is a thick layer of less cross-linked peptidoglycan. We have a keratin spore coat. So warm, moist, nutritious conditions can cause the spore to germinate. And we'll get an active spore. So when we take a look at bacterial chromosomes and plasmids, okay, um, we have a lot of loop domains, and we're not going to get into this too much because the bacteriophage can reproduce by any number of mechanisms depending on its structure. So let's take a look at this. So these homologous recombination between two bacteria, they have to find a homologous zone to begin recombination and exchange DNA. Rec-A is essential for this. So remember we talked about the pilus on the bacteria, right? That is used as a sex pilus through conjugation to meet with the other bacteria and insert its chromosomal contents so that that bacteria will change and produce more. Site-specific combination is integration of DNA. It's an addition. It's a fertility factor where the bacteriophage into a bacteria, movement and insertion of transposons. So there's all different kinds of complex ways that bacteria can multiply. And you can have two different bacteria that multiply and cross genus and species. So if we take a look, the male cell, known as F, due to its fertility factor, has sex pili, genes that stabilize the pair, genes that direct DNA transfer, origin of transfer, and insertion sequence. The female, they don't have a fertility plasmid. At the end, the cells become F plus donor. So here's the conjugation pilus, here's the male plasmid, here's the female cell. So they're going to transfer its material from the male cell to the female. So the donor cell attaches to a recipient with its pilus, and the pilus draws the cells together. And it forms a cross bridge, or a mating bridge, is what they call it. Here, the cells connect with one another, and then one strand of plasmid DNA is transferred into the recipient. The recipient then synthesizes a complementary strand to become an F-positive cell. The donor synthesizes a complementary strand, restoring its complete plasmid. How cool is that? You see what just happened here? Female, nothing. Male, plasmid. Sex pilus, mating bridge. Mating bridge complete. DNA goes in. Female cell is able to manufacture circular DNA, and then the male cell will replenish it. So transformation, and that was conjugation, by the way. In transformation, the cells become able to bind short pieces of DNA and import them into the cell, and these are incorporated by homologous recombination. So let's take a look at this. You have free DNA from a dead cell. Here's your bacteria chromosomes. Non-competent, rough, non-encapsulated streptococcal pneumonia. Growth factor conditions are optimal. Nasal pharynx, oropharynx. So the competent cell now is able to bind DNA. What happens? They uptake these little guys in the surrounding fluid, and then they stabilize some of the genes by homologous recombination. So in this circle, do you see how we have new incorporation of the free DNA from one of the dead cells? Now we have smooth encapsulated and pathogenic strep pneumonia. Boom. We didn't kill it, and it got stronger. It used transformation. So if we take a look, looking here, we have the cell tries to give all of its DNA to F, but stabilization doesn't last. The F cell stays as the recipient and needs homologous recombination to express its new plasmids. So here's our cells. Here's a mating bridge. The chromosomes are F factor pilus and bacterial genes. All right, and then he replenishes his internal genes, and then this is what we have for the female. But in conjugation, we have to use the dead cells, the free DNA from dead cells, to bring it all together. So eventually this guy will make what this guy makes. So lysogeny is a state of stable association of bacterial and viral DNA. It may impart new characteristics to the bacteria, such as lysogenic conversion. It continues as long as the repressor gene is functional. So if there's no, how do you say it? If there's no resistance, the bacteria will just continue to take over, right? So think of co-beds as when two people share a bed and someone gets a little bit pregnant by a phage, such as cholera toxin, O antigen of salmonella, the botulism toxin, erythrogenic exotoxins of strep pyogenes, diphtheria toxin, or the Shiga toxin. So now we're going to look a little bit at how the antibiotics fight against these bacteria, because bacteria get smart. They do. They get smart. They said, oh man, I've been getting killed by, beta-lactam penicillins. I better make something so I don't get killed, and they make beta-lactamase. Now we have to invent another medication to kill the beta-lactamase or prevent the beta-lactamase from destroying the antibiotic. A good example of that is augmentin with potassium cavulate. It's the potassium cavulinic acid that destroys the beta-lactamases. So we have penicillin and cephalosporins. The production of beta-lactamase, cleavage of beta-lactam rings, is the mechanism of resistance of the bacteria. Aminoglycosides, production of acetyltransferase, adenosyltransferase, or phosphotransferase, inactivation of the drug by acetylation. Chloramphenicol, production of acetyltransferase, inactivation of the drug by acetylation. Tetracyclines, bacteria are able to cause the medication to actually efflux and pump it right out of the cell. Sulfonamides active export out of the cell and lowered affinity of the enzyme. Vancomycin ligase produces cell wall pentapeptides that terminate the D-ala D-lactate, which will not bind to the drug. Tricky, aren't they? Kind of scary, right? Minimal bacterial inhibitory concentration. So what does that mean? It means how long can the drug survive? At what concentration? And hopefully we get no growth. So RNA or DNA can be icosahedral or helical. Typically, your DNA is helical. Here's your herpes virus. It's got a lipid bilayer and glycoprotein outer layer. This is a nice drawing. It kind of looks like a Where's Waldo drawing. But this kind of goes into, how we get infected and how we get protected. So attachment of 1 is prevented by a vaccine. 2 is penetration. 3 is uncoating. 4 is macromolecular synthesis. So early messenger RNA and protein synthesis, replication of the genome. The DNA goes to the nucleus and the RNA goes to the cytoplasm. Late messenger RNA and protein synthesis. 5 is post-translational modification, which usually happens on the ER, goes to the Golgi body apparatus and is exited through the cell. Assembly of the new virus particles, and then release and lysis of the cell, or budding or fusion, where it just kind of comes out. So the virus HIV targets T helper cells, macrophages, and microglial cells in the brain. Receptor on the host is CD4 plus CCR5, macrophage, and CXCR4 T cells. Epstein-Barr, its target is B cells, CD21 to CR2. Rabies is the neurons. It's the acetylcholine receptor. Rhinovirus, common cold, are respiratory epithelial cells, and they use ICAM1. So this kind of goes over how the viruses replicate. This is the retrovirus, HIV. And it's kind of complicated. We have drugs for HIV that affect a lot of the different processes. So what happens is double-stranded DNA plus the virus, they don't need an intermediate. They go to messenger RNA. We take a single-stranded RNA plus the virus. We get single-stranded RNA with a negative sense of the virus, and so it must bring in RNA polymerase inside the viron. The goal is to make double-stranded RNA from a DNA particle, so they reverse engineer it, and we get a retrovirus. The exceptions is hepatitis B, parenteral genome is double-stranded DNA, unless single-stranded DNA is used to make double-stranded DNA. So they get very smart. Viruses get very smart. So the process of disease, the process of infection. Acute infection, we start. Look at smallpox. Smallpox has been eradicated due to the vaccines, but smallpox will become acute and you either recuperate or die. Simple as that. Which is now because of we've had the vaccine for so long, we've tried to stamp it out entirely. Here's measles. Acute infection. Sometimes we get rare complications. Intact virus is not made. We can still get post uh, panencephalitis of the brain. Latent infection, chickenpox. We've got vaccines for that. Many times kids got the chickenpox. They'd get better. And then it would go into latency in the dorsal root ganglia. And later on in life, it would come back as shingles. Chronic infection, this is Hep B or C, and this is the viral shielding. Although there is medication now that can help treat Hep B and C. Age is important for the diagnosis of viruses. So infants, we're looking at respiratory sentinel bronchiolitis, parainfluenza or croup, rotavirus, which is infant diarrhea and influenza. In utero, we test the female for the cytomegalovirus, rubella, HSV2, HIV, and parvo B19. This can all happen in utero. Don't necessarily test for parvo B19, but if a woman gets it while she's pregnant, it can be devastating to the fetus. At birth, hepatitis B and HSV, herpes. Children, we look at for rubella, parvo B19, measles, mumps, hepatitis A, epidemic gastroenteritis or the Norwalk virus, and varicella is chickenpox. Adolescents and young adults, herpes simplex 2, HIV, Hep B. I don't know, they don't have mono or Epstein-Barr in here, they should. And adults, whatever, pneumo, mycoplasma pneumonia. Senior citizens, herpes zoster, St. Louis encephalitis, West Nile virus. So, markers, hepatitis B surface antigen. Okay, think about it. Hepatitis B surface antigen. This is not protective. This is not good. It's found during acute diseases and persists in infection. More than 6 months, if this guy's still around, it's chronic. Hepatitis B surface antibody. Well, that's good. We have an antibody that our body made. It provides immunity to the hepatitis B vaccine. Hepatitis C antibody. Typically, your immunoglobulin IgM plays an important role indicating recent infection if we have high GM levels. It's useful during a window period, and IgG later on, 8 to 12 weeks later, usually is a sign that the patient has recuperated from the infection because IgG is an immunoglobulin made by B cells and plasma cells that are actually specific toward that particular virus. Now, hepatitis E antigen, it correlates with active viral production and infectivity. Hepatitis E antibody basically just suggests a lower risk of transmission. So here's acute hepatitis B, okay? So let's see what happens. This is hepatitis core, is red, green is hepatitis E antibody, and orange is hepatitis antibody. Okay. So we see here's acute infectivity. All right, this person is highly infected. They can pass it on. But notice how the infectivity drops, right? It just drops. While it's dropping, other things are happening, right? Your body's producing antibodies. C is an antibody. HBE is an antibody. And the surface antibody is here showing remission. Chronic, on the other hand, means that they're always going to possess the infective, they're always going to have the virus, and it's always going to be infective in their body. It could not be as strong as far as, the actual virus goes, but they still have it. And it can wax and wane, but they're always chronic. So this is, well, we're looking at AIDS now. So the HIV, okay, this is important. So everybody, can be at risk if you're not careful. Let's look at HIV. Okay, so here's the viral RNA genome. It's RNA. The nucleocapsid, the reverse transcriptase, the integrase, the protease. P24 antibodies are detected for diagnosis. We can do that. Rapid HIV test. Reverse RNA transcriptase produces double-stranded DNA provirus. How smart is that, right? GP41 and GP20, okay, these guys divide and separate. They fuse to the host cell. This is a transmembrane glycoprotein. GP120 is a docking protein. It attaches and interacts with our CD4 helper T cells and co-receptors. This is how HIV gets into the cell. Decreases CD4 and MHC1 expression, required for progression to AIDS, responsible for virulence. This is the NEF gene. It's very complex. And again, very smart. So if we take a look, these are some of the diseases that people with AIDS get once their CD4 levels drop below 200, and it's pretty serious. Normally, you and I will get these. If we have healthy CD4 counts above 500, normally we are able to fight off many diverse, very odd diseases. But pneumocystis jirovecii, we start prophylaxis, we start treating with TMP-SMX, usually at around less than 200 when the CD4 count drops. We discontinue it once it becomes over 200 again, within 3 to 6 months. And probably a good question you should be asking yourself is why do people with HIV, why does their CD4 count, wax and wane? Why does it go down? Why does it go up? And a lot of it has to do with their bodies are over, and Cryptococcus neoformans less than 50 continue. Okay. So fungi, this is Malaysia furfur, spaghetti and meatballs. This is Coccidioides immitis, spherules and endospore. And they're showing you where different fungi are endemic. Coccidioides is the southwestern states, blastomycetes, dermatitis on the eastern part of the United States, Midwest, and then Histoplasma capsulatum. So this was a lot. I hope it helped a little bit. Enjoy, keep studying. See everybody later.